Um, I'm not a very good speaker, and normally when I stand before people, I freak out. But I'll try to to be a little confident today. And thanks, Matt, for that wonderful introduction. It was a little bit too long than the ones that I sent to Kelly. Apparently, she got more from our website. I'm glad to be in the town of Condor. I've never been here before. It's an exciting little place. And thank you, Matt, for welcoming us to your town. And I also want to say that I'm not a specialist in cyanobacteria. I'm just interested in it. And when Joey contacted me, I was like, how did you know I'm interested in cyanobacteria? Because I don't have any information out there that I'm a specialist in cyanobacteria. But anyway, I have an interest because I met one fine gentleman from Pine Lake, and he's interested in cyanobacteria, and that made me want to know more about cyanobacteria. I got a lot of stuff here for you. As I said, we all have our own understanding of what blue-green algae is. I know everybody knows, and we have our own ideas. So consider me as not a specialist, as just one of you who was requested to put some information together about cyanobacteria. And I want to start with, cyanobacteria, they're part of microbes, like Lisa was talking about when you think of E. coli, all those fecal coliforms, all those bacteria out there, they're all microbes. And most of the organisms that we have on our planet, they are actually microbes. And very few of them have been characterized. We really know little about them. And some of them play a very essential role on the planet, on the environment. But only a tiny bit of them, very little of them cause harm as pathogens or producing byproducts like cyanobacteria that I'm interested in that produce toxins. So what are cyanobacteria? Most people think cyanobacteria as being algae. They've been classified as blue-green algae. People think of them being plants, but actually they are not. They are not plants, they are not algae, they are bacteria. But they just have similar characteristics as plants, meaning they contain chlorophyll, which enables them to produce their own food. And because they have chlorophyll, and then in the presence of nutrients, then that's when they multiply. And when they multiply, they form blooms. And once they form blooms, that's now we start freaking out. Like we don't like the look of our water. It looks green, and then that's when you get all the advisories about that it's dangerous to go to the lakes. And again, there's one point there that not all blue-green algae are green. Are blue. Some are yellow-green. Some are green-yellow. Some are even red. So they are all different colors of the cyanobacteria. And there are cyanobacteria present in all the lakes. They are present, but they only become a problem during blooms. And once that blooms occur, in most cases, during the presence of excessive nutrients, most of these nutrients additions into the lakes, into the water bodies, are being blamed on human activities in the watershed. And because of that, when you have cyanobacteria blooms, then they pose health risks to humans and livestock and wildlife. And most typically, these blooms happen during the summer when we want to use the lakes. And just one point there, that it's not possible to tell cyanobacteria apart from algae with the naked eye. You just can't go into the lake, scoop some water from the lake and say, there is cyanobacteria. You may see the film. You know, some of them are colonial, meaning that they stick together like Lisa was talking about, a biofilm. You might see that film, it's because they are colonial. But otherwise, maybe mention it somewhere, but you can't go and lift cyanobacteria from a water body like you can some other algae. And some of the factors that cause blooms, fertilizers, general eutrophic conditions, and you see now in a few slides that most of the lakes in Alberta, they are actually eutrophic. Naturally, they have a lot of nutrients in the lakes. So, or when you have the low nitrogen to phosphorus ratio, that favors bloom formations, or when you have bright sun and calm conditions in the water body, that will also favor bloom formation. Also, when you have changes in the zooplankton grazers in the water body. These might happen especially if you have introduction of new species into a water body. And on the N and P nitrogen and phosphorus ratio, it's been found that when the ratio is less than 10 to 1, that one has been shown to favor the heterocystus blue-green algae. And the significance of that is that these are the ones that, because they have the heterocysts, that is a site of nitrogen fixation. And we've heard about cyanobacteria and eutrophication. Cyanobacteria blooms, for example, I'm sure you saw some of these cases where a few animals were killed in Manitoba because of the cyanobacteria blooms. And obviously eutrophication is just the enrichment of our water body with nutrients. And there were some experiments that have been carried out in Ontario for a long period of time, where they were looking at isolating some sections of a lake, and then they did different nutrient additions to different parts of the lake to see which of the nutrients, nitrogen or phosphorus, contributes to cyanobacteria blooms. And they found that phosphorus is the main culprit of eutrophication. And just to summarize, what happens on a given watershed, you have all those, you have discharge of untreated municipal sewage all the way from farms, just runoff from erosion, and I mean, there are so many sources of nutrients into any given water body from a watershed. So you can see that there's just lots of them. And as I mentioned, I went to the website, the Alberta Environment and the Sustainable Resource Development, just to see how are the lakes classified. And this is the map that I found, and they have different colors, from those ones which have very little nutrients in the lake, to one medium, to the ones eutrophic, which have lots of nutrients, or to the hyper-eutrophic. And they have, if for example, you go into their website, they have classified the lakes according to the nutrient concentration in the lakes. And they have the oligotrophic, which have very little nutrients. And in these lakes, you have little possibilities of having blooms. Also, even the mesotrophic lakes. But when you come to the eutrophic and hypereutrophic lakes, these are the lakes where you find a lot of cyanobacteria blooms in the summer. This is where our pine lakes, pigeon lakes, all these lakes that you heard about having cyanobacteria blooms in the summer, this is where they fall. And so what does this classification show us? It just tells us that water quality is impaired by increased biological activity, and this biological activity obviously is influenced by how much nutrients are available in the water body. And as I mentioned, just by addition of nutrients, it's not enough. It has to include an increase in phosphorus. So phosphorus has been determined as the main culprit. And I just tried to research around and see what kind of actions are being taken to control these. And I found that Alberta Environment and the Sustainable Resource Development, they've been doing a lot of monitoring from the 1940s of many of these lakes. And there is data out there about the conditions in the lakes, I think year in and year out. And they have been, I think that's where the lakes, ALMS, Alberta Lakes Management Society, they've been supported by that Alberta Environment to do most of these data collections. And so there has been some collaboration with those kind of organizations and also with watershed groups. For example, the Pine Lake Restoration Society, where they installed the phosphorus project, the removal project, about 15 years ago. So, so my interest, as I mentioned, is on cyanobacteria blooms, and why are these blooms such a bad thing? And they may, obviously, they make our water look unattractive. Nobody wants to go and swim in green, filthy-looking water. But actually, you can have a bloom, but still, if you are brave enough, still go and swim in it. If there are no toxins. You know what I'm trying to say? You might look unattractive, but you might still be able to go and swim in it, unless there are toxins in the water. And remember that not all cyanobacteria blooms lead to an increase in toxins in the water, because not all species of cyanobacteria actually produce toxins. And like I mentioned, toxins, they are amplified by the activities that take place on the watershed. Otherwise, there are normally lower levels of cyanobacteria toxins in the water body unless the blooms are elevated. I just found some examples of harmful algal blooms. I thought I'll share with you some examples this is from marine habitats where they found some sea lion deaths linked to toxic algal blooms. Some researchers in University of California, Santa Cruz, they traced some toxins from algal blooms, and some cases where consumers were advised not to eat some spot harvest shellfish and crabs and those other organisms because of the fear of toxins. And also in freshwater systems, there was places in Texas where thousands of fish were dying because of toxins produced by cyanobacteria, or where there was ingestion of toxic material was implicated in the death of a pet dog in Washington. And if we move, also in other cases, I'm originally from Kenya, so I had to look for a case from Kenya, so where some lesser flamingos deaths were attributed to cyanobacteria toxins, or some where liver failure was also attributed to the exposure of toxins produced by cyanobacteria in Brazil, or in this case in China, where toxic blue-green algae was suspected for the high rates of liver cancer at locations, or some locations in China. But it's just not those far away places. Coming closer home here, we've seen that somewhere. I found an example in toxic algae bloom was found in an Alberta lake. It was in CTV a couple years back. Or those Wizard Lake, Pigeon Lake, there were some advisories given. Or if you are from Pine Lake, from around there, and there's an algal bloom prompts a warning that we should not go to Pine Lake. That's what got my interest. That's why I started developing this interest in cyanobacteria, because of that algal blooms in Pine Lake. So you probably will see signs. I didn't see a sign posted, but in other places they post signs like no swimming or boating because of toxic algal blooms, or in some lakes these algal blooms then they just create this filthy mud on top of the water body. That's a case from Minnesota. Like I mentioned, we think of algae as being that green algae, but actually there are different types of algae. So we have the golden browns, blue greens, the dinoflagellates, and diatoms. So I looked for some information on each one of them to see if there are any culprits in each of these groups that produce toxins. And I found that there are lots of, like I mentioned, there are lots of species of blue-green algae, but these are the main ones that produce toxins. So you have the Anabina, Anabenopsis, and all those ones. So these are the main genus of blue-green algae that produce, so far, that are known to produce toxins. And on the right-hand side is the type of toxins that they produce. And on the brown algae, these are some of the species that are thought to produce toxins. And on the dinoflagellates, there's also a long list of dinoflagellates that have been found to produce toxins. And even when you look at diatoms, I don't know if you know about diatoms, but there are some species of diatoms that produce toxins. But when you read about the newspapers, all the news that comes to us, what they always say is that cyanobacteria produce toxins. So you just think that in any lake, it's only cyanobacteria that produces toxins. But actually, all these other species too produce toxins. But they produce the toxins in, I think, in small levels that do not reach the level that an advisor will be posted. An advisor will only be posted when you have a bloom. And most of the bloom, as I mentioned, occurs with cyanobacteria. That's why toxin production is normally associated with cyanobacteria. So, like I mentioned last year when I had about cyanobacteria, so that's what raised my interest. So, and I found that the toxins that cyanobacteria produces, they have been linked to health problems. And like I mentioned, how Joey found me out, I crafted this little proposal where I want to go out, hopefully if I get some funding, if you know people out there and you're interested in cyanobacteria, you know funding agencies, tell them about me, that I'm interested to do this research, so that I can determine what are these toxins that are found in this lake. And my interest, my background, was mainly in fisheries, because I read somewhere that actually the toxins from cyanobacteria can actually be incorporated in fish tissue. And from fish tissue, they can be transferred to human. For example, when you go out there fishing, like right now I know people are doing ice fishing out there. And if the toxins that were produced by cyanobacteria were taken out by the fish, and then we went and we got the fish and we ate it. So I wanted to see if that toxins will be transferred from, I'm interested to find out if the toxins in the fish can be transferred into human beings. And obviously there are several pathways by which these toxins can be transferred to human beings. It's either by eating contaminated food, drinking contaminated water, swimming, so that they go through your skin, or it can be through inhalation. And like I mentioned, there are cyanobacteria. There are about 2,000 species, and 40 of them are known to produce toxins. And so we know those species. So, and my interest will be, in addition to what kind of species of cyanobacteria do we have for now in pineapple, because that's where my interest is. And so we will try to determine if there's anabina, there are microcystis, planktrix, or not, all those species, whether they are in the lake. And I've been talking about toxins, and the toxins that are produced by cyanobacteria, they are called cyanotoxins, and some of them are neurotoxins affecting the nervous system, or they can be hepatotoxins affecting the liver, or some of them are cytotoxins or gastrointestinal and dermatotoxins. So there's a wide range of toxins that are produced by cyanobacteria. Don't worry about those formulas. I just felt like just putting them there. I don't even know what they mean, but I just thought they look beautiful and I put them there. So examples of some neurotoxins, it's like anatoxin A, which I found that it mimics acetylcholine A. Those are also other terms. Never mind about them. Just know that they produce neurotoxins, and there are different types of neurotoxins that are produced. And also there are hepatotoxins too. And as I mentioned, these cause liver damage and can lead to liver cancer. I'm not trying to scare you about the toxins. They are not that serious. It's not a big issue. I just want to make that one clear. Yes, there are toxins. They can lead to cancer. They can lead to liver cancer. They affect the nervous system, but you really have to drink a lot of that water with those toxins to really be affected. So, like we saw that example that we saw in Brazil, where it was contaminated with microcystins, and a lot of patients died. Because I thought, since this presentation will be on file with the Alliance, just in case somebody wants to go and get more information, there's a slide here which lists the toxins and the references where you can find more information about that. There are a couple slides there. So there's a lot of unknown things out there that we need to work on. For example, why are certain species of cyanobacteria producing toxins just only in a few occasions? I found some information that they don't produce toxins all the time. They don't produce toxins on a few occasions. Also, be interested, I found if these chronic diseases will actually caused by cyanotoxin. I won't probably do the research, but I'm trying to research on that by reading what people have found out. And what causes these algal blooms? I know we say that when you increase the nutrients and you have a shallow water body, a stagnant water body, then phosphorus loading, then you have blooms. But probably there are other factors that we can also consider. And how can we remove the toxins that are produced from cyanobacteria from the water? And what is the exposure rates in Alberta? I don't know. I haven't had if people have actually been exposed to the toxins and been affected, but I would like to find that one out. So I haven't done that yet. But like I mentioned, cyanobacteria blooms, they are related to nutrient increase in the water body. So I can make a few recommendations on how I can control that. And there are three aspects. Thinking of prevention, increasing public and government awareness, and obviously continuation of research and monitoring. On prevention, we're interested in how we can prevent cyanobacteria blooms and how to prevent human exposure to cyanotoxins in the event that we have blooms. So those are the areas that I'm interested in that we could work on. And when we have, in case we have these cyanobacteria bloom preventions, I'm thinking where there should be prioritizations especially in those lakes where there have been occasions of blooms in the past. And one area that I'm thinking about is the enhancement of buffers. Obviously, we all know what the effectiveness of buffers around our water bodies and even the activities that that take place in the watershed. And I'm also interested in constructed wetlands. At Olds College, we do have constructed wetlands. And we're thinking about running a bunch of experiments, and some of them probably, if we get some funding, might be related to cyanobacteria toxins. And just to talk a little, to add one more slide on enhancement of wetlands. It's possible, I was just thinking that it's possible we could have wetlands around these water bodies, but probably their holding capacity for nutrients has been reached. So that when nutrients have been released, you think you have, we haven't even removed any of the wetlands around our water bodies, but they are not serving their purposes because of lots of prior release of tons of nutrients into the system, such that the wetlands that you have cannot take up any more of the nutrients. So just thinking about wetland preservation, could even constructed wetlands work? I don't know, but if that's a possibility, probably to only be a temporary fix. But that's some of the areas that we can look at. To prevent exposure, obviously we need to increase our treatment, water treatment methods and technology and provisional guidelines. And there are guidelines out there, especially that have been put forward by the World Health Organization. And also we need to increase public awareness, things like meetings like these ones where we can come and share ideas. So that's important. Let's all get connected, be informed, and where action is needed, we need to demand that action be taken. And obviously we also need monitoring, and especially in those lakes, on those water bodies where we've had cyanobacteria blooms for a while. And because I realized from reading most of the lakes that receive the advisories are almost the same length as you see advisories each year in, each year out. And also look at those areas which have had cyanotoxin exposure before. And with the advancements of technology, we could think of remote sensing, because from aerial photography, I know these lakes are small bodies, but at a good magnification, you'll be able to detect the different types of blooms how they take place during the season. And that one can be shown by the changes of the color. And also research needs. We need to have certified standards, consistent sampling protocols, so that when we go to a particular water body, we use the same protocol. As you'll see in a few slides, it all matters where you collect your water from. That's very important. Also, we also need some long-term studies to identify the key environmental factors, and also find out methods for early detection, and with that we can probably develop some predictive models. So I have, for example, here where I mentioned consistent sampling protocols. Sampling location in a lake matters. For example, if you look there, it's not spread out consistently in a given water body. I don't know if you can see that there was, from this study, there were variations by orders of magnitude. This is water collected from the same lake, but from different locations, where one has, for example, 13 micrograms per liter microcystin, while the other one has just 4 micrograms of microcystin. So it's important that we develop those protocols. And same again with consistent sampling, we can use plankton, or whole water sampling, or filtered sampling, so that from there you have the total toxin that can be found in the water, which is the dissolved phase and the toxin that is in the particulate form. For example, what I'm saying there is where they found that when you just used plankton nets, they underestimated the microcystin concentrations. And so, like I also mentioned about the early detection and predictive models, this one can give us the real-time variables, specific conductivity, pH, temperature, all those ones, because those conditions you can predict from the conditions that you have in a water body when you're going to have bloom, as you have the changes in those ones. For example, this is just like one of them. You see the temperature, as the temperature changes, it also influences the concentration of oxygen in the water body. Although you'll see that temperature increases, and then thereafter you have a decrease in oxygen in the water. So with with technologies like these, then we'll be able to monitor our lakes so that we'll be able to predict when we're going to have the blooms. And some of the available research techniques out there, we have what are called the bioassays. Enzyme-linked, for example, the enzyme-linked immunosorbent assays for ELISA, which can be used to assess the cyanotoxins. Or we could use the gas chromatography method. Each of these methods is listed with its advantages and disadvantages. Or we can use liquid chromatography. We also use that to measure the cyanotoxins. But it's been found out that it's better when a combination of the methods are used, because they found that, for example, some of the microcysts that will be missed by liquid chromatography or mass spectrometry can be identified when ELISA was used. I thought I'd give you some examples of some interesting findings that have been found when dealing with toxins produced by cyanobacteria, where they found that they exhibit a wide range of toxicities and toxic effects. So that's from one research in '99, and also they also found that the peak microcystin values may even occur in the winter in some lakes. You know, when you think you don't think that there are some bacteria in the winter, probably, but they have been found that the concentration of the toxins, the peak, was identified in the winter. And also, as in some research, they also found that the seasonal patterns, they are relatively consistent between years in some lakes. So you always have a spike at a given time of the year. And in some, they have found that these microcystin to be correlated with factors that influence cyanobacteria growth, which makes sense. And there are a ton of examples there where they try to correlate the association between microcystin and environmental variables, and they found it's not direct, it could be complex. And in some, they found that microcystin is not strongly correlated with measures of cyanobacteria community, where an increase in cyanobacteria or a bloom does not necessarily lead to an increase in the amount of toxins that are in the water body. And also, some of the correlations were found to be linear between the toxins, which, for example, an increase of chlorophyll, which indicates that there has been an increase in algal growth, either algae or cyanobacteria. So what I've tried to show you here is that the cyanobacteria produce toxins, and various methods that are out there that I personally am thinking of trying to use, especially as I collaborate with some of you when you have these problems in the lakes. And for example, like I mentioned, right now I'm thinking about collaborating with Pine Lake Restoration Society. And all of us want to use our lakes, and we don't want them green. If there's something that we can do in the watershed that can prevent the cyanobacteria blooms, then we can collaborate as communities, as societies, so that we can prevent those blooms. And nobody likes getting advisories about their lake being sick. Nobody wants that. So, yeah, I'm interested in collaborating with each one of you if you are interested. And obviously, there are lots of resources that are available there that talk about cyanobacteria and the problems that we are all experiencing and how we can control those problems. And that's a picture of our constructed wetlands. If you have not come to see them at Owens College, please do come, and then we can talk more and see what kind of projects that we can work over there or in the various watersheds.